When a patient walks into a clinic with a swollen, painful knee joint, the visible swelling is not the beginning of their disease. It is the endpoint of an invisible biological process that has been running for days, weeks, or even years. That identical outward symptom could be driven by three entirely distinct origins: a bacterial invasion of the joint space, a misdirected attack by the body's own white blood cells, or sharp uric acid crystals depositing in the tissue. In pathology, the initial trigger of the disease, whether a bacteria or a crystal, is called the etiology. The step-by-step biological cascade that unfolds inside the tissue as a result is the pathogenesis. A clinician who attempts to treat the visible symptom without identifying the underlying pathogenesis is guessing. Prescribing antibiotics for an immune attack or immune suppressants for a bacterial infection allows the actual disease process to continue unchecked. To make accurate clinical decisions, medical professionals have to move past symptoms. They must master how biological systems fail at the cellular level and how we systematically classify those failures into treatable categories. Almost all systemic disease originates at the microscopic level. It begins when a single cell encounters a stressor it cannot handle. A functioning cell needs continuous oxygen to produce ATP. Hypoxia, oxygen deprivation, is the most common cause of injury. When oxygen drops to zero, ATP production halts. The sodium-potassium pumps on the membrane lock shut. Water and calcium flood in, and the cell rapidly swells. If the stressor is removed quickly, the injury is reversible. A footballer collapsing from heat exhaustion experiences widespread cellular swelling, but with rehydration and cooling, the cells pump the fluid back out and recover. Prolonged stress, however, pushes the cell past the point of no return. This microscopic failure scales directly to macroscopic death. In a myocardial infarction, a blocked artery cuts off oxygen to the heart muscle. Within minutes, millions of starved cardiomyocytes undergo this hypoxic cascade, swelling and dying. The resulting heart attack is the macro expression of cellular hypoxia. The first pathway for cell death is apoptosis. It is a clean, controlled self-destruction program where the cell shrinks and fragments symmetrically. This is essential for normal biology, like sculpting fingers during embryonic development, by eliminating the web of tissue between them. During apoptosis, the cell packages its DNA and internal debris into tiny membrane-bound fragments. This contains the damage, allowing neighboring cells to quietly digest the remnants without alerting the surrounding tissue. The second pathway is necrosis. This is a chaotic, strictly pathological form of cell death caused by severe injury. In necrosis, intense swelling stretches the cell membrane until it physically ruptures. The cell violently bursts, spewing internal enzymes and debris into the environment. The most common subtype is coagulative necrosis, which occurs in solid organs after ischemia, a severe loss of blood supply. The architecture of the dead tissue is preserved for days, but the internal proteins coagulate and solidify, leaving behind a firm, lifeless mass. The distinction between these two pathways is a critical trigger for disease. While apoptosis is silent, the chaotic chemical spill of necrosis sets off the body's alarm system, sparking a destructive and powerful cascade known as inflammation. Acute inflammation is the body's highly stereotyped, immediate response to necrotic tissue or bacterial puncture wounds. It is a rapid defense mechanism designed to isolate and destroy threats. The response begins in the blood vessels. Histamine causes local capillaries to dilate and their walls to become highly permeable. Blood flow increases, and a protein-rich fluid called exudate leaks into the surrounding tissue. This vascular shift produces the classic physical signs of inflammation: redness, heat, and swelling. This vascular leak acts as a delivery system for the cellular phase. Neutrophils, specialized white blood cells, squeeze out of the leaky vessels and flood the injury site. They engulf bacteria and necrotic debris, neutralizing them with toxic oxygen species and digestive enzymes. This illustrates what happens when acute inflammation fails. If a pathogen like Mycobacterium tuberculosis is too resilient for neutrophils to destroy, the immune system shifts tactics. It deploys a secondary wave of defensive cells, macrophages and lymphocytes. Unable to kill the bacteria, they pack tightly together to form a dense, spherical barrier called a granuloma. Inflammation evolved to protect us, but when an inflammatory response cannot clear a threat and transitions into a chronic state, the continuous cycle of tissue destruction and attempted healing replaces functional organ tissue with rigid scar tissue. Disease does not strictly require an external pathogen like bacteria. Often, it originates from internal system failures, particularly within our immune and genetic programming. In a type 1 hypersensitivity reaction, the immune system aggressively overreacts to a harmless external antigen, like penicillin or a peanut. It misidentifies the substance and triggers massive histamine release, dropping blood pressure and swelling the airways in a life-threatening mechanical failure known as anaphylaxis. Autoimmunity is a different kind of malfunction. Here, the immune system loses self-tolerance entirely. It misidentifies the body's own healthy tissue as foreign and destroys it. In type 1 diabetes, for instance, immune cells systematically hunt and kill the insulin-producing beta cells in the pancreas. Failures also originate at the base code of biology, genetics. A single typographical error in a DNA sequence can alter the structure of a vital protein, completely derailing its function. In sickle cell disease, a single amino acid mutation causes hemoglobin inside red blood cells to polymerize when oxygen drops. The once flexible cell physically distorts, hardening into a rigid crescent shape that wedges into small blood vessels and blocks blood flow. When genetic mutations strike the specific genes that regulate cell division, the result is neoplasia, the loss of normal growth control. If a tumor suppressor gene fails, damaged cells are allowed to replicate indefinitely, accumulating further mutations and forming malignant tumors. Whether a disease is driven by a mutated gene, an overactive immune cell, or a rogue replicating tumor, the mechanism always leaves a measurable footprint. Clinicians must trace this footprint backward to identify the cause. Understanding the isolated mechanisms of cell injury, inflammation, and mutation is required, but it is not enough on its own. To process thousands of possible conditions, clinicians overlay a structural system of classification. This structural grid represents a matrix grouping system. It categorizes diseases by overlapping orthogonal variables. The primary x-axis represents etiology, or the cause of the disease. This groups conditions by their specific origin points: infectious, genetic, neoplastic, or iatrogenic, meaning physician-caused. The primary y-axis tracks the clinical course, or duration. It separates rapid-onset acute conditions from prolonged long-term chronic states. Additional informational filters are then layered onto this grid, identifying the primary organ system the disease affects and its communicability, whether it can be transmitted from person to person. A single disease does not sit isolated in one category. It occupies multiple coordinates simultaneously across this matrix. Tuberculosis, for example, plots perfectly at the intersection of an infectious etiology and a chronic clinical course, while also filtering accurately as a respiratory and communicable disease. Identifying a disease's exact coordinates on this matrix dictates the treatment. It tells the clinician whether they need an acute surgical intervention, a long-term quarantine protocol, or lifetime metabolic management. A theoretical matrix is clean and orderly, but clinical medicine is unpredictable. Applying these definitions is what separates delaying symptoms from saving a life. Knowing that acute appendicitis is driven by severe inflammation explains why the tissue must be surgically removed before swelling causes necrotic rupture. Conversely, categorizing sickle cell disease as a genetic autosomal recessive condition means treatment goes beyond pain management. It dictates the necessity of preventative genetic counseling for the patient's family. Throughout a medical career, the specific names of diseases and the pharmacological tools used to treat them will evolve, but mastering how cells fail, how tissues react, and how those reactions are classified provides the permanent intellectual scaffolding required to diagnose and treat the human body.